So my PhD is all about empathy for another's pain. And if we try to define empathy in a very easy way, we can say it's sharing as well as understanding the emotion of another person. So we feel what the other person feels. Of course, the working definition that we have in our lab is a bit more complex. So we define empathy kind of more as a process consisting of multiple different components. So first of all, we have to either directly observe or imagine the emotions of another person. For example, you see your work colleague hitting his or her elbow on the door frame while coming into your office. Second, we have to be in a state of isomorphic affect sharing, meaning we have to feel the same feeling that the other person is feeling. So we would also feel some kind of pain in that moment. And thirdly, we have to have some kind of self-other distinction. This means that we know the source of the emotion lies in the other person and not in ourselves. And we have to distinguish this. This is also very important in order to avoid personal distress, so that we feel stressed out by the emotion that we are feeling right now. So this definition kind of combines more automatic bottom-up, but also more controlled top-down processes. And it also differentiates empathy from other mechanisms, such as emotional contagion, compassion, or sympathy. And in my PhD, I focus on this isomorphic affect sharing component and the relationship between the first-hand experience and the empathic experience by focusing on somatic pain. But why should we do this? Why is empathy relevant? I think that there are a lot of reasons why feeling empathy and why investigating how it is processed in the brain are important. So first of all, we are inherently social beings. So be it in friendships, at work, or in romantic relationships, we constantly have to process our surroundings. We have to interact with other people. And a very important part of these surroundings are dealing with emotions. So both positive and negative, or neutral, of course, both coming from our own body or coming from other people. So empathy, in a way, can help us process these emotions, respond appropriately in social situations, and it can also help to foster bonding. The majority of social neuroscience research has used somatic pain as a model to investigate empathy. There are a lot of good reasons for that. So one, pain is something that can be very easily and robustly be induced in psychological experiments. So if I give someone a shock, it will hopefully feel painful all of the time and every time. So this is nice for us. And second of all, there's already very strong evidence regarding the neural networks underlying pain. So it is very nice to use it as a model to study empathy. But before I explain the network of pain processing in more detail, I want to give you a quick explanation what pain actually is. So it constitutes an unpleasant sensory and affective experience associated with actual or potential tissue damage. And as you may have noticed, this definition does not necessarily need to be limited to first-hand pain. So it could also include other types of pain involving other people, for example, social exclusion or just seeing other people in pain. And I think that this also, for me at least, encompasses empathy. So just as a little warming up on the topic of empathy, I'm going to show you 3 pictures, and I just want you to look at those. You don't need to say anything now. Maybe you can write some associations in the chat and we can have a look at them later, or we can just discuss it later. So maybe just reflect on how you perceive these pictures and your initial reaction. Okay. And these are, by the way, some stimuli that I'm going to talk about later in one of the tasks. So now let's talk about first-hand pain. So of course, pain is considered to be a very complex, multidimensional experience. And usually it's divided in 2 major components. The first one is the sensory discriminative component that is more the basic representation of pain. So for example, the intensity, the location, the timing, or physical characteristics. And it is represented in the brain in primary and secondary somatosensory cortices here in green. And then there's the more affective motivational component that codes more for the subjective aspect. So how we judge this pain that we're feeling. Is it unpleasant? Do we need to withdraw from it? And it is represented in anterior and mid-cingulate cortices in red and anterior insula in brown. So if we now look at how people process the pain of others, there are of course lots of differences to the first-hand experience, but also some similarities. So here in the figure we see in green what is active during first-hand pain, and in red we see the overlap between what is felt during empathy for pain and first-hand pain. So there seems to be some common things happening in the brain. And indeed, it has been shown many times in the past that receiving pain and observing others in pain recruits overlapping activation in these two networks that I just presented. And mostly this overlap occurs in regions coding for the affective component of pain, but some studies also showed an overlap in somatosensory areas, and they really highlight the importance of this component. And this has been taken to suggest that shared neural representations may underlie both of these experiences. This means that we might recruit the same mechanisms when we feel pain ourselves and when we see someone else in pain. However, it is unclear whether and how we can infer that shared activations, so brain activity that we measure with fMRI, for example, are equal to shared representations, so the underlying networks or functions. So if we actually recruit the same mental processes during empathy as when we directly experience the emotion that we're empathizing with. And this gap has recently been addressed, well, recently in 2015 by our lab and with the lead author Markus Rüttgen in the so-called placebo empathy analgesia studies. And here the authors employed a more causal approach than what had been done usually before by manipulating the experience of first-hand pain using placebo analgesia. And then they looked whether this also had an influence on empathy for pain. I'm just going to shortly explain the experiment that they did. So half of the subjects were led to believe that they received a pill that had a strong analgesic effect. In reality, it was a placebo, so it had no pharmacological effect whatsoever. But they thought that this would reduce their own pain. And then participants went into the scanner and they had a task where they received electrical stimulation on their hand, and they also observed another person receiving such stimulation. And interestingly, the placebo was not only able to reduce the self-perception of pain, as we would expect, but it also reduced ratings related to empathic responses, so observing another person receiving the stimulation. And it was visible on the brain level as well in the form of reduced activity in insular and anterior mid-cingulate cortex in the placebo compared to the control group. However, so to sum up this a bit, some empathy studies find overlapping activation in the affective motivational component. Others find an additional involvement of sensory motor or somatosensory areas. And the placebo analgesia study that I just presented reduced activity only in the affective component, so anterior insula and AMCC. However, this is a bit interesting because the manipulation, so placebo analgesia on its own, usually affects both pain components, so the affective and the sensory component similarly. And this mismatch could have resulted from the choice of paradigms that was used previously that could maybe have prevented the observation of such a modulation of the somatosensory component. And here, empathy research often makes a distinction between cue-based versus picture-based paradigms. So in the top right, you see kind of an example of cue-based paradigms where abstract symbols or facial expressions show in a more indirect way who is now getting, for example, an electrical stimulation on the hand. While in picture-based paradigms that you see on the bottom right, we have more visually explicit stimulus material where there are body parts, so hands or feet, that are in painful situations, for example, a needle being stuck in the hand or the hand being in some kind of pain. And so we could hypothesize that somatosensory components might only be involved in empathy when empathy is instigated by visual cues. So when the attention is more explicitly directed to the location or the type of pain, because this is what the somatosensory component is also doing. So coding for these more basic aspects. And these previous placebo analgesia studies, they used a more cue-based approach and facial expressions of pain. So they did not use picture-based approaches to try and find an involvement of the somatosensory component. So therefore, in this pre-registered study, we wanted to know whether placebo analgesia can also interfere with the sensory discriminative component of empathy for pain by using two tailored tasks that were modified from previous empathy for pain paradigms. So this is just a quick overview of the session. Participants came to the MRI scanner together with another person, posing as a second participant, but who was in reality a confederate of the experimenters. And then we calibrated the pain of each person individually for each hand, and then we conducted a placebo analgesia manipulation. And I will explain this more in the next slide. Just to give you an overview, after that, participants then completed two pain tasks that were, as I said, specifically tailored to observing a possible involvement of the somatosensory component. So we calibrated average pain ratings for high, medium, and low pain individually for each hand, because it can be that the pain ratings vary per hand. And then we conducted a localized placebo analgesia induction, where a study doctor came in and applied gel on either hand, but on the right hand, the doctor said that this gel was a strong local anesthetic that would reduce their pain on the right hand, while the left hand just received a normal basic skin cream. And as in previous studies, the gel both gels were the same or nearly the same and had no active pharmacological compound. We then also did a conditioning procedure to amplify the effects of the placebo in accordance with previous studies. And here we told participants that they would receive high pain intensity stimulation on both hands. And on the left hand, this is also what we did. But on the right hand, we covertly lowered the stimulation intensity to medium pain to suggest a substantial relief of pain by the placebo or by the medication. And just so you have kind of an idea what the study setup looked like in our scanner. So here's, for example, me posing as a study doctor, explaining the procedure to a participant. Here we have kind of roughly the way that we applied the electrodes. Of course, we used more medical tape and made it a bit more clean, but this is just an example. So we had a lot of cables going on in the scanner. And then this was our setup in the scanner, where the participant was lying on the right side in the scanner. And then on the left side, we had the confederate sitting. And of course, what is not shown in the picture are all of the cables, because there were 4 cables per participant. So the confederate had cables as well, just to give the impression that he or she was really receiving pain as well. So we had some criteria to identify non-responders to the manipulation that is usually done in placebo inductions. For example, if participants had really strong doubts about the manipulation and about the placebo, so they thought, yeah, this is a placebo for sure, or they didn't believe what the doctor was saying, things like that. Or if their belief in the effectiveness of this gel kind of decreased very strongly over the course of the session. And we had fixed criteria, and we kind of grouped participants after each session, whether they were a responder or a non-responder. And we pre-registered that we would collect data until we had reached 45 subjects who had a strong first-hand placebo analgesia effect. So this is what we have. In the end, half of those people were male, and of course they were mostly students around 24 years. And importantly, all of those people were very strongly right-handed. This is what the first task looked like. This is an adaptation of the task that was used in the placebo analgesia studies by Markus Rüttgen and colleagues, but we modified it to make it a bit more somatosensory focused. So we did have a cue-based setup in the sense that there were cues being shown on the screen. So whether the stimulation would be painful or non-painful, who would get the next stimulation, the self or the other person. But then in addition to that, we had pictures of either the participants or the confederates' own hands from an egocentric perspective shown on the screen, so participants could focus more on the location of pain. And then after receiving electrical stimulation, participants rated either their own or the other person's pain, as well as their own unpleasantness to the other person's stimulation. We did 2 manipulation checks to make sure that we really had a first-hand placebo effect going on, as this was very relevant for our research question. So I asked participants 3 times during the session how much they believed the medication to be effective. So how effective do you estimate the gel on your right hand to reduce your pain in the task? And I asked this right after the gel was applied, then after the conditioning procedure, and then also after all tasks were completed and participants came out of the scanner. And as you can see here, we observe a significant increase in belief through the conditioning procedure, so from pre to post conditioning. Interestingly, the belief also decreased significantly until the end of the session. However, it did not decrease lower than the first rating that we had pre-conditioning. So to me, it's still a very good indication that even though the placebo effect might have decreased a little bit over the session, it was very high in the middle, and then it did not decrease super low in the end. And maybe that's interesting for you to know. So all of the tasks were done right after the conditioning procedure. So when the belief in the placebo or in the medication was highest. For the second manipulation check, I asked participants after the session how painful on average the shocks felt on each hand. So how painful was the stimulation on the right hand with the medical gel and on the left hand with the hand cream. And here we also found a significant difference. Of course, this is kind of a rough estimate participants did after the session, but still nice to see that they subjectively felt that the pain on the right hand was lower than the pain that they got on the left hand, even though they got the high intensity stimulation on both hands that we had calibrated before. So if we now look at the results of the first task, so from now on, you will always see the placebo, the right placebo hand in orange and the left control hand in blue. So if we look at the first hand pain ratings, we can see that the electrical stimulation participants received on their placebo hand, shown in orange, was rated significantly less painful than the stimulation on the control hand, shown in blue. So this is a nice, let's say, third manipulation check that the placebo induction worked quite well. But this is also no surprise because, of course, we chose placebo responders. So yeah, this is basically what we expected. However, if we now look at the 2 empathy ratings, so how painful was it for the other person and how unpleasant was it for you to watch the other person getting a shock, we can see that this effect did not transfer to empathy. So the pain and unpleasantness ratings were comparable for the 2 hands. We then conducted region of interest analysis in primary and secondary somatosensory cortex, as well as in the 3 affective regions that were also done in the previous studies by Rüttgen and colleagues to investigate a transfer of this effect on empathy for pain. Today, I will focus on the somatosensory brain regions because this was our main research question. However, for example, in S1, we did not find any modulation by the placebo manipulation in S1, neither for self nor for other. And in S2, we did observe evidence for a first-hand placebo analgesia effect in right S2, so as you see on the right side here, where activity corresponding to the placebo hand was lower compared to the control hand. But again, we found no transfer of this effect to empathy in a localized way. So to sum up the results for task 1, we did observe a strong localized placebo effect during first-hand pain in behavior and also on the brain level, especially in right S2. But empathy for another person was not influenced by a localized pain reduction in a specific body part. And we also did not observe evidence for somatosensory sharing in S1 or S2. For the second task, we made the stimulus material even more explicit and more strongly focused on showing the location of pain. And these are also the pictures that I showed you in the beginning, where I asked you to reflect a bit on your own pain experience. The task was as follows. So we had 2x2 design, where pictures of either the right or left hand were rated, while this hand was either in pain or not in pain. And here we adapted previous picture-based paradigms where the hands are in everyday painful situations. For example, the hand being burned on a stove, or cut while gardening, or hit with a hammer, things like that. And importantly, the location of pain was always the back of the hand, because this was where we had applied the placebo gel. So it was important to us that the stimuli also reflected the same location. And this is how we made the stimuli. So as you can see, here's me with my hand in a paper cutting machine. And this was actually the most dangerous picture that we took, because I had to simultaneously try not to get cut by the paper cutting machine, but also make the picture as realistic as possible. In the end, this picture didn't make it in the final stimuli set because it was too unrealistic. If we look at the results of this task, again, we had the 2 empathy ratings, how painful for the other person and how unpleasant for you. We also did not observe a transfer of the first-hand placebo effect to empathy for everyday painful situations. So no differences between the ratings for right and left hand. We conducted the same region of interest analysis also for this task. And here, again, similarly to the first task, we did not observe a transfer of the placebo analgesia effect to activity in S1 or in S2. Interestingly, and this was really paradoxical or interesting to us because we hadn't expected it, we observed evidence for an opposite effect in the 3 affective regions that we had as region of interest, where post-hoc Bayesian analysis showed substantial evidence for an opposite effect, especially in bilateral anterior insula, where the brain activity for stimuli corresponding to one's own placebo hands or right hand stimuli was higher compared to left hand stimuli. However, this finding will have to be followed up because it was not part of our pre-registered predictions, and we just noticed it basically in the end. So this will have to be investigated more clearly in the future. So to sum up task 2, placebo analgesia did not reduce picture-based empathy in terms of subjective ratings in a location-specific way, and also did not reduce activity in affective and somatosensory brain regions in a location-specific way. But we did find, as an exploratory result, increased brain activity in bilateral insula for stimuli corresponding to one's own placebo hand. So to conclude, empathy for pain may rely more on sharing another's affective compared to the somatosensory state. So the general processing of another's pain might have had a higher weight in our task than the processing of the exact location or source where that pain is occurring. I think my studies also show that it is very important what task you choose when you investigate empathy, because whether you choose a cue-based or a more picture-based paradigm can lead to very different results, and this influences what conclusions you can take. And in sum, I think that I try to refine the understanding of the neural underpinnings of empathy for pain, which is important when we try to think what magnitude of influence our own pain experience has on empathy and how the two are connected. I want to thank my lab and my 3 co-authors, Friederike Riva, Markus Rücken, and Claude Lamm. And if you have any questions, I would be happy to answer them. Thank you.